Hello and welcome to Chapter 2. Congratulations, you made it through Chapter 1. That was a pretty hefty chapter. You're in luck. Chapter 2, short, sweet, to the point. Chapter 1 was the introduction. I had to show you everything we're going to do. But Chapter 2, we just get into the fine details of how we're made, basically, in utero. So developmental. So it's 35 slides. Let's get started. Chapter 2 is outlined here, so if you're following along in your book, it's Nolte page 39. You can see that at the bottom. So we're going to talk about pretty much everything that's on this outline is a test question at some point in time or another. It's a matter of what test questions I pull from the ones that I've written. Your boards will also pull very similar test questions. So I would know all of the things that are part of the outline in this, and I believe the next slide as well. So neural tube gives rise to the central nervous system. Neural tube, central nervous system. Sulcus limitans separates sensory and motor areas of the spinal cord and brainstem. Now don't worry, we're going to have pictures of these later. This is just the outline slide. Okay? Neural tube has a series of bulges and flexures. There are 3 primary vesicles and 5 secondary vesicles. The growth of the telencephalon overshadows other parts of the nervous system. The cavity of the neural tube persists as a system of ventricles. The neural crest and cranial placodes give rise to the peripheral nervous system. So neural crest, cranial placodes are peripheral. Adverse events during development can cause congenital malformations of the nervous system. Think folic acid deficiency and neural tube defects. Defective closure of the neural tube can cause spina bifida or anencephaly. An in front of a word tells you without. Encephal is brain, so without brain. Defective secondary neurulation can cause a distinctive set of abnormalities. The forebrain can develop abnormally even if the neural tube closure is complete. Okay, so that was pretty heavy, so I'm going to define what all of that meant, but as you go back and study those specific slides. Those first 3 slides, and you should be able to get that once you get the meaning behind it. So let's talk about the development of the nervous system. An understanding of the development of the nervous system helps make sense of adult configuration and organization. So what do we think about here is neuro rehab. So when we're doing our neuro rehab, we want to think about, well, how was this person made in the first place, right? So how did we figure out how to myelinate in the first place? Babies are born not fully myelinated. That's why they can't run and jump and play and talk and do advanced things like you can when you're an adult. So how are we going to instigate healing of the system? We mimic it by how does it learn in the first place? So a lot of our neuro rehab looks like games that we play when we're babies in order to develop. Developmental aspects, our embryology here. So our primary germ layers, you might have heard this in quarter 1. So we have our from superficial to deep. We're looking at ectoderm. Okay, so ecto, you want to think of things like the nervous system and skin. Mesoderm, middle. Okay, so middle in muscle is the way I remember that one for the M's. And endoderm, endo within, so that's pretty much everything else and it's deep. Okay? Formed early in embryonic development, specialized to form the 4 primary tissues, so our nerve tissue, muscle, connective, and epithelial. So those 3 germ layers, that ectoderm kind of looks like the mustache. Remember when that was all the rage, to wear mustaches on things? Ectoderm, epidermis, and nervous system. Meso, everything else. Endo, air, and gut. Another picture for you here. So pause at any of these pictures that you like to kind of give yourself a better understanding of it. Another one here for you, giving you specifics: ecto, epidermis, nerve, meso, notochord, muscle, kidney tubules, red blood cells, endo, pancreatic cells, thyroid, and lung. The neural tube and neural crest give rise to the central nervous system and peripheral nervous system. So you can see how it's formed on the right there. So the neural plate, neural groove, neural fold, neural tube, these are all central nervous system. So the neural crest is peripheral. So when you have to memorize something like this, which is really just strictly memorization to be able to answer a test question right. So I get that, I know that, but it's your boards that do that to us. So that's why we have to do it here at this level. So neural crest cells, peripheral nervous system. That's what you want to remember. Anything else, central nervous system. Okay, so neural crest, peripheral. So those are going to be our neurons, our glia, the peripheral nervous system, melanocytes, chromaffin cells of the adrenal medulla, and then our notochord, which is going to be our primitive backbone here as well. So neural crest, peripheral nervous system is the one that you really want to remember out of these things. So this is just kind of a play-by-play of formation here. So you can see number one, we see the neural groove and the fold. You can see that notochord underneath where that backbone is going to be. And we call that groove and fold the neural plate. And then you can see it kind of starting to fold in. That groove is becoming a little bit more apparent. We could see the crest starting to form on top there, like crest of a wave, think. And then you could see the fold. You can still see the notochord there in all of these, 1, 2, 3, and 4. So 3, you could see it just a little bit more. 4, you could see where the skin has come up and over. You could see where there might be a problem in that stage. That would be spina bifida if we didn't see that coming up and over as easily. Primary versus secondary. So I'm not going to ask you anything about primary and secondary neurulation except for this right here. Secondary, I even bolded it and underlined it because I've seen similar questions when I took some of the board review books out from the library. I saw one on secondary neurulation, and I thought it was kind of a random question, so I want to make sure that you guys see it. Secondary, sacral, S for S. Secondary is sacral. Spinal cord forms after the neural tube closes. A secondary cavity extends into the solid mass of cells at the caudal end. Caudal meaning tail, remember. Sulcus limitans separates sensory and motor areas of the spinal cord and brainstem. So sulcus limitans separating sensory and motor. That's what we want to know, and that's happening at the cord and the brainstem. So in the brain itself, we know that that's the central sulcus that's going to separate sensory from motor, the pre and post central gyri. So don't get it confused with that. This is spinal cord and brainstem is the salient feature here that we want to pay attention to. So white matter, we are calling that the marginal layer. The mantle layer is going to then be the gray matter. So remember, what makes white matter white is myelin. So mantle layer, gray matter. So we're looking at the alar plate being the posterior horn, and that's going to be sensory. The basal plate is going to then be the anterior horn or the ventral, which is then going to be responsible for motor. And then we're going to see ependema here, and that is lining the central canal, and that is remember those kind of hairy-looking teeth, the ciliated ependymal cells that are making cerebral spinal fluid for us. That's what it was talking about. Here's a picture of the sulcus limitans. I even put a little purple arrow next to it so you can see it. So sensory and motor. Note that it is not the midline. It is the line off to the side from the midline, denoting sensory from motor, not the actual midline. Neural tube has a series of bulges and flexures, so it's just talking about how our head is kind of flexed there, and then we see the telencephalon is going to become part of the cerebrum there, and then we could see the cephalic flexure, that kind of head area. How our brain gets to be in that kind of weird convoluted shape is all it's saying. And then we'll look at that picture on the right, the prosencephalon, mesencephalon, rhombencephalon, and then teldi, mesmet, mai. Is you can see mesencephalon didn't differentiate over that blue middle part. That's still mesencephalon on the right as well. So we'll look at that right here. So the way I remember this is prom-rom. So P-R-O-M-R-H-O-M. So if you look at 1, 2, and 3, prosencephalon, mesencephalon, rhombencephalon, prom-rom. If you just take the first couple letters. That's how I know those are my primary vesicles, and in order from top to bottom. They're going to differentiate or kind of bump out, grow out into what we call the 5 secondary vesicles. We're going to call these, the way to remember it is tel, di, mesmet, mai. Kind of rhymes, has a little rhythm to it. Tel, di, mesmet, my. I didn't make that up. That's just a neuro thing. I'll let you know when I make stuff up, so I don't leave you hanging out there in the world saying weird stuff. But it's tel, di, mesmet, my. Okay, so prom rom, I think I did that one. So, but it works. And then tel, di, mesmet, my. Telencephalon, diencephalon. Mesencephalon is the same from the primary. Okay, so note that those are easy test questions. Which of the primary vesicles doesn't differentiate out? Mesencephalon. And then we call it metencephalon and myelencephalon. So when you see myelo, that usually means bone marrow or cord. Okay? So deep within is what that's talking about. You could see this on Nolte page 41 and Manter and Gatz pages 12 and 13. It's confusing at first, but you do it a couple times and you get it, and then you'll get those test questions right. They're easy test questions once you get it down. Here's a little chart telling you. So there's our prom-rom, right? So prosencephalon, mesencephalon, rhomencephalon, the primary vesicles. Who's that going to become? So the way that we do this is mesencephalon does not differentiate. So we just go straight across with that mesencephalon. Okay, so when we write it out, it's just boom, mesencephalon, fine, right? And then our little tel, di, mesmet, my. So we're like, all right, tel, di, well, mes is in the middle, and then met, my. Cool. All right. So then our primary vesicles, there's 1, 2, 3. So I know that my tel and my di have to be above that mesencephalon, right? So tel, di, mesmet, my. And then met and my are below it. So there you go. It just kind of works that way. And then what's above it? So we see that number 2, we're going to just kind of follow that line again. Mes means middle, so that's midbrain. Super simple. And then that cerebral aqueduct in between the third and the fourth ventricles is kind of this middle line that we look at first. So memorize that first. And then once you start to really know your anatomy better throughout this term and next term, you're going to just know that the lateral ventricles, the third ventricle, are above the cerebral aqueduct. You're going to know fourth ventricle and the central canal are below the central or cerebral aqueduct. But you might not know that now, and that's okay. Okay, so we're just going to start small here and build, and don't panic. Okay? But above midbrain there, on that neural derivatives, just to the left of the cavities there, on that you look at midbrain and you're like, all right, so we know where our midbrain is. So we have medulla pons midbrain. Okay, so that'll get drilled into our heads over and over again as we study this. So here's midbrain. So we go below that, and there's medulla, there's pons. And next to pons is cerebellum, so we're okay with that. Then midbrain, all right, medulla, pons, midbrain. And then everything else is above that. Okay, everything else is above it. The cerebral hemispheres, thalamus, hypothalamus, retina, obviously that's the eye, and other structures. So the way to really start to memorize this is look at that middle one and then go above and below. All right, let's get a little more specific. Here's the telencephalon, so tel, di, mesmet, my. Telencephalon, again, cerebral hemispheres. Okay, so the cerebrum, what you think of, if I asked you to draw the brain on the first day of school without knowing anything about it, if you didn't have any background in it, you would probably draw the cerebrum. So lateral ventricles, the caudate nucleus, putamen, globus pallidus, that's going to be more basal ganglia, or what we call basal nucleus now. Amygdala, we associate that with anxiety, and then our hippocampus here, memory, spatial orientation, things like that. So telencephalon, these basic structures here. Diencephalon, the thing you want to see here is that it is all things that have the word thalamus as part of the word. Thalamus, hypothalamus, epithalamus, subthalamus. And then there's retina and third ventricle. It doesn't really fit. But if you see thalamus in the word part, it's diencephalon. Looking at that epithalamus, so stria medullaris, pineal gland, and posterior commissure. Pineal gland is probably familiar. That is what takes serotonin and methylates it into melatonin, or helps you become sleepy in the presence of darkness. Mesencephalon, there's that middle again. Okay, so that's midbrain. So it's not that simple though, right? So midbrain, we're going to differentiate that into an area called the tectum. Okay, these 4 bumps on the area called the tectum. We have 2 superior colliculus, or superior colliculi, and then inferior colliculus. Superior colliculus is part of the visual reflex, so it's responsible for if you see something out of the corner of your eye and you turn and look, superior colliculus is telling us to do that. Inferior colliculus is if you hear something out of the ordinary and you turn and look. Inferior colliculus is in charge of that. Note that both of them end with turning and looking. That's the one where students can get tripped up on the test question. It's what is the stimulus? The stimulus for superior is vision or sight. You could go S for S, sight. Okay? And then inferior is hearing. Tegmentum is more anterior to the tectum. So tegmentum is going to have the red nucleus and substantia nigra. Substantia nigra just means black substance. So when this black substance is pale, we know that the patient probably has Parkinson's disease. So this is melanin that is making it dark. Okay? So melanin that's making it dark. And it is either a byproduct or has something to do with the production of dopamine. So they're not exactly sure the exact steps there. It's either like the point of it, right? Or it's like the ash. If you had a fire and then the ash that's left over, is that melanin creating the dark substance? Or is the dark substance like the point of it? Not quite sure in neuro right now. And if we were sure, we'd probably be able to figure out Parkinson's a little bit better. The reticular formation is here as well. Third and fourth cranial nerves, remember, are here. So our rule of fours with the cranial nerves works out here. So you number them 1 through 12, and then you just go medulla, pons, midbrain, and above. So on each 4. Okay, so 12, 11, 10, 9, that's going to be medulla. Okay, and then the next 4 is going to be pons, and then the next 4 is going to be midbrain is 3 and 4, and then 1 and 2 are further above from that. At the base, we're looking at the cerebral peduncles and the cerebral aqueduct. You can see the pictures over there. Here's the pons. Okay, so rule of 4s again are cranial nerves, and then pathways to the cerebellum. So it's really just this nice pathway wrapping around to the cerebellum. Cerebellum, you want to associate that with balance. Superior cerebellar peduncle, middle, and inferior peduncle is just a stalk. So these are just pathways to and fro the cerebellum. And then another part of the metencephalon is part of the fourth ventricle. Part of that fourth ventricle is going to also be in the myelencephalon. Okay, so part of the fourth ventricle, part of the central canal. You can see cranial nerves 12, 11, 10, and 9, and their nuclei. And then in the medulla, we have the olives and the pyramids. Okay, so olives and pyramids, just structures of the medulla, and we'll get into that. It's kind of a cool picture. You don't have to memorize this. I threw it in there because it was kind of neat to see as we're talking about developmental. You could see a 13-week-old embryo all the way on up, right? And the system of what does the neural tube look like, and what does the system of ventricles look like? This is what the ventricles are going to look like in our full-grown brain here. So I'm going to point out those two kind of ram's horn-looking things. Those are the lateral ventricles. And then you can see the directional horns there, anterior, lateral, and inferior horns there as well. And then there's the third ventricle and the fourth ventricle. Note there is not a 1 and a 2 ventricle. It's just 2 lateral ventricles. And then a third ventricle midline, and the fourth ventricle is midline as well. So another heading on that outline was this: the neural crest and cranial placodes give rise to the peripheral nervous system. So neural crest becomes peripheral nervous system. Cranial placodes form the sensory components of the peripheral nervous system of the head. Adenohypophyseal placode forms the anterior pituitary. That's a mouthful right there. So when we look at adenohypophyseal, that's code basically for anterior pituitary. So we'll see neurohypophysis, and that's posterior pituitary. So you'll get that in endocrine, A for A, adeno, anterior pituitary. Adeno actually tells you that it's glandular. Okay? So. And then the lens placode, lens of the eye. That makes sense, right? So this is Nolte pages 48 and 49, and then I cited the picture there for you as well. Some of you might ask, what's a placode? That's probably not a term that we hear that often outside of developmental neural world, right? So it refers to ectoderm thickenings in the cranial region that have important roles in development of special sensory and systems, and I pulled it from that citation there. It's kind of a cool looking picture there. So note the optic placode or eye vision apparatus there. Nasal placode, frontal nasal. You can see maxillary, the front top part of the teeth area. Mandible, lower down. So kind of a neat way to look at that. And then where the heart is going to be, the liver. Okay. And just another picture here, kind of help you out with what are the placodes here. So I don't test you off of this. It's kind of detailed. So developmental problems, you'll see this on your boards. It's not an uncommon test question. What's spina bifida occulta? Occult means hidden. Okay, so occult means hidden. So failure of the posterior arch to join, but you won't necessarily see it. You might see it on x-ray. So when I was in chiropractor school here, my student doctor, the one that was taking care of me when I was in first quarter and she was in ninth quarter, told me that she had a spina bifida occulta. Didn't even know it until they took x-rays at chiropractor school. So it made no difference to her, really. Meningeoceal, outpouching of the dura. So remember, our P arachnoid and dura are our meningeal layers. So outpouching of the dura and CSF. Myelomeningioceal, so myelo is telling me that it's the cord, and meninge is telling me that it is the meninge. So remember, P arachnoid dura. So this is an outpouching of the dura and the cord here. So, and we're going to see that cerebral spinal fluid in there as well. So when you see seal at the end of a word, it means tuber or hernia. So you could see that this would be this kind of mass or herniation of it if you look off to the right. The difference there, the meninges, okay, versus spinal cord and meninges. So on the furthest one to the right, you can see the cord in there. Really, it's cauda equina, right? It's not the cord in there, but it's fine to call it that for this. And you could see the cerebral spinal fluid in there. You could see that the meninges are affected as well, the dura. The meningeoceal is just the dura and the cerebral spinal fluid, but it looks like the cauda equina is in its normal place. And then you could see the spina bifida occulta. You could see maybe just that bone didn't form all the way. And then there's a little tuft of hair that might give a clue that there's something wrong there or something slightly abnormal there. And then the baby up at the top, that's quite obvious. That's either meningeoceal or myelomeningioceal, and that obviously needs surgical attention. So this is an ultrasound of the meningeoceal. Don't worry, I will never test you on this. I'm not quite sure what I'm looking at either. People that can read ultrasounds are magic in my eyes. But you can see the cord here, so you can see the vertebra, you could see the brain, so just kind of an interesting picture there. All right, that's it. So that was chapter 2. I told you it was short and sweet. It's kind of like a reward for getting through chapter 1, right? So here's your questions here that you want to look at. So number 1, a stillborn infant exhibited severe facial abnormalities, and upon autopsy, it was discovered that the brain lacked significant gyri and sulci. At this point, the most likely diagnosis would be what? What is that defining? So look those words up and then define it. Number 2, during the second trimester of a pregnant 24-year-old, abnormally high levels of alpha-fetoprotein are found in her blood. Such elevated levels may be indicative of what fetal abnormality? Again, look up those words, define them, and you will find your answer. Number 3, failure of the metencephalon to develop correctly will have the greatest effect on what? 4, failure of proper neural crest migration will have the greatest impact on? So pause at any of these that you need to and make sure you can answer. Number 5, failure of the rhombic lips to form properly will most likely lead to problems with what? So why do I include these in here? Because the book included them in here. So your boards uses Nolte as a reference in order to take test questions from. So I think it's a good idea to look at what test questions Nolte comes up with for you to take. The end. So this is where I took some of that information from. I told you it was short, sweet, to the point. Go take your Brightspace quiz for your attendance verification and for a couple points there. And then don't forget your lab video and your lab quiz as well. And happy studying, and I'll see you for chapter 3.